So thank you everybody and Human Society for inviting me to be here today presenting my work. So I'm going to talk a little bit about our working in my lab, modeling some diseases from the central nervous system. So in my lab we work with autism spectrum disorder. We work also with Duchenne muscular dystrophy. And the last year we start to work with Zika virus to model the microcephaly, as students also did. And today I'm going to present some results on autism spectrum disorder. So, to the ones who don't know nothing about autism, autism is a disorder. It's a neurodevelopmental disorder that we have the children who present autism, they present some deficit in the communication, social interaction, and also in the behavior. The behavior is typical behavior for autistic people is with stereotyped movements, for example, and also they have deficit in cognition. They have cognition impairment, and also the prevalence is very high. You have 1 to 68. This is in the US. But around the world, we can say that we have 1 to 2% of the population manifests some kind of autism or some symptoms of autism. So it's a lot of people with that. And we have many ways to study autism, right? So we can use post-mortem brain, which is a good model. It's hard to say something like that, but it's important to donate our organs for research. So this is really important. We have some contributions really relevant on that, but when we study the brain is a fixed tissue, right? So it's that. So we cannot have any insights about the communication between the cells. So it's important, but it's a stuck model, right? So we also can use genetics to study autism. So it's important to say that we have more than 1,000 genes related to autism. So it's a kind of black box, but it's important, but doesn't bring any insights directly related to the neurological developmental. So we have also animal models, but here we have to make a comma, because as I told you before, we have many genes related to autism, right? And we are talking about an animal, so the background is completely different. So we don't have any genetic background in an animal model related to autism. We can make autistic mice, but they are not with our brain. So we have a problem. How do you study a brain in a dish without doing brain picking, especially for autistic people? I'm not talking about the IPS, but since then we can do mini brains in a dish, and we do this in our lab. And it's for autism is really relevant to study, to use IPS to model autism and to do the brains, the organoids in the dish, because every brain is unique, right? So the genetic is unique. So if you consider that you have a black box, it's important to keep this black box in your dish, right? So that's why we use IPS to model autism. So to do that, we just use patients and also the controls, and then we model them and compare to see which are the differences or which we have in common. So the first group that used IPS to model autism was a group in the University of California in San Diego from Alison Muotri, the ones who Troy mentioned in his presentation. And they model Rett syndrome, which is a kind of autism. I would say that they took off from the umbrella of autism because they're on a genetic disease, right? But the people who has Rett syndrome, they present symptoms on autism. So That's a simple model to study autism. And they have really important insights doing this kind of approach. In addition, they used the neurons in a dish to test drugs. And they discovered one drug which is particularly important, rescued some neurons, and this discovery make a clinical trial start in US. So we have this clinical trial on Rett syndrome using IGF-1 because of this work. So this is a model to show that we can use mini brains in a dish to test drugs. We don't need animals. In this case, it's especially important for autism to use the brains in a dish. The second work from the same group, they used one patient, one autistic patient. I mean, it's a typical idiopathic autistic patient. They found a gene after that. This patient had a deletion in a big part of a chromosome, which disrupts this gene, TRPC6. And they found that using hyperforin, which is a drug used for depression, they could rescue the neurons. So this is the second example of a good job using IPS to model disease. And this is the third article related with autism. We just have 3 published until now using this technology. And in this work, I could participate in this work. They used 6 autistic patients, all of them idiopathic and with microcephaly when they were born. They used these patients to understand a little bit more what happens with autistic patients, especially the ones who were born with microcephaly. And they found that the proliferation of the networks of the neural cells were completely altered compared with the controls. So also they used the same IGF-1 and they could rescue the neurons. So based on that, my lab is working with this IPS model. But before that, we started we tried to see if we could use another model, not the fibroblasts, because all of these 3 works were using fibroblasts, biopsies from fibroblasts. And then we start to work with cells from the dental pulp. So we remove, we wait until the thing to follow. Yeah, you can remove, the dentist can remove, but we don't do that, but it's possible if you want. We just wait until the teeth follow, and then we remove the pulp, and then we reprogram the cells, and we could make very good neurons and astrocytes and whatever. So because of that, the work was published, and because of that, we create a project in USP called the Tooth Fairy Project. So it's an old joke, Steve's not the joke, but I'd like to say that the Tooth Fairy exists and lives at USP. So we have this project running there since 2009, and we receive donations from children from all over the country, from autistic, from Duchenne muscular dystrophy, and also from controls, people without any kind of neurological disease associated. So because of that, we create a NGO called the Tooth Fairy Project. We have a homepage, and also we have a page on the Facebook. This is just to collect it's a channel to talk with the families and to receive the donations. So until now, we have more or less 300 cell lines. We have more than 300 donations. But we have now, we got 300 of cell lines from autistic patients in our biobank. So it's the first biobank of autism in Brazil. And we are proud that we receive donations from all over the country. And based on that, we work doing the disease modeling. This is in a dish, so this is the workflow. And I'm going to show you some unpublished results regarding our autism research. So this is Fabielle Russo. Fabielli was my PhD. Now she's my postdoc, and she worked in her PhD with 3 idiopathic autistic patients. So we make the IPS from the cells from the dental pulp. We make the IPS. We also karyotype the cells before we go on to make sure that we don't have any kind of imbalance in the chromosomes. And then we make the NPCs, and until this step, we couldn't see any differences between the controls and the patients. And then when we make the neurons, we start to see something different. Well, let's show you something. Okay, so we quantify the synaptogenesis on these neurons. So this is the way we do that. And we use a program to do that. Let's see if this video is going to... So we use this program to quantify the synaptic buttons. So we call the puncta, which is the pre and post synaptic together. We do separately and then together also. And then we count them considering the branches, I mean the length of the branches. So this is very specific. And then we can quantify if we see some differences between the patients and the controls. We noticed that the controls, compared with the patients, with the autistic patients, they have more, the control have more synaptic numbers, could be pre and post synaptic buttons compared with the controls, and also the puncta number, the autistic present less synapses, so there is an impairment in synaptogenic process. We also use Maestro. It's a multi-electrode array, the ones that David showed you before. So we plate the neurospheres in our plate, and then we can measure the communication between the cells in many 12 channels at least. So we could notice that autistic patients, they presented less spike rates compared with the controls, as you can see here. So this could explain a little bit about the cognitive impairment, for example. Also, we measure glutamate, which is a very important neurotransmitter, and we notice that autistic patients, they present less glutamate compared with the controls. And also, we produce the cells from the glial cells, the astrocytes. We produce them separately from the neuron, our mixed population of neurons. This is just the characterization of the cells. And we notice that the astrocytes from autistic patients, they produce more ROS and more glutamate compared with the controls. And also we measure some cytokines, and one special that I'd like to call the attention, it's IL-6. We noticed that autistic patients, they present more IL-6 than the control ones. And this is particularly important because IL-6 is, for the central nervous systems, it's very toxic, right? So we did from this part we did a Coculture experiment. So we produced astrocytes separated from the neuronal mixed culture population, and then we just sorted the neurons, and we plated astrocytes and neurons on top of the astrocytes, and we make some different conditions to see that. So here in this picture you can see the astrocytes, and on top of the astrocytes one neuron that we put on top of these astrocytes. So in this slide, the astrocytes are in black. I didn't call the attention to the astrocytes, just to the neurons that are on top of these astrocytes. And then you can see we mix the conditions. So we use wild-type neurons with wild-type astrocytes, and we have this kind of morphology of the neuron. When you use autistic astrocytes, I would say autistic, right? It's not autistic, but just to understand. And autistic neurons, we have this kind of morphology, more simple. And when we just use the normal neuron with autistic astrocyte, we have this kind of phenotype. So the neuron become more simple with less branches than we saw before. And when you use autistic neuron with wild-type astrocyte, we have a kind of recovery here. So this means that maybe autism, we just look at the neurons, but maybe there's something on the astrocytes that we should pay attention. Not only the morphology, we also count the number, the puncta again. We also try to measure the synaptogenesis. And just looking to this graph, which is the controls, so we could notice that when you use a control neuron with an astrocyte from an autistic person, we have less synaptogenic process, and the opposite is true, right? So if we just put a good astrocyte, we have recovery of the number of synapses. Not only the morphology, but just the synapses, we also have recovery. Thinking about the IL-6 and trying to see if we could do something related to the IL-6. IL-6 is very important, especially because it's IL-6. The IL-6 level is high when you have people with refractory seizures. So we try to see, one of our patients has a lot of seizures. So we try to see if we could block this at some point and see what happens. So we use, this is the control, so we put recombinant IL-6 in the control. So we have control astrocytes and control neurons, and the number of synapses just diminishes. And also when we have the patients producing a lot of astrocytes with the neurons and also astrocytes producing IL-6. When you treat with anti-IL-6, we have the recovery of the number of synapses. So this means that IL-6 impairs the synaptogenesis in autistic neurons. Of course, we combine all of this data with the genetics, so we know exactly which genes are impaired in our autistic patients. But in that point, this is not relevant because we have the patients are genetically different, right? But they just, even being genetically different, they have the same clinical symptoms and they have the same biological phenotype when we use this methodology. So this is something interesting to comment. So as a take-home message for this part, I could see that autistic neurons are less functional. They undergo less synaptogenic process. Healthy astrocytes can recover the neuronal functionality, as I showed you. IL-6 blocking could rescue ASD neurons. Yes, and that's it for the autistic part. Just to, I'm not going to say a lot about Zika modeling, but just to show that we use the same methodology using the Tooth Fairy project, but with the controls to model Zika, and we published last year. I forgot to say, these are my three students that did the job. Fernanda Cúbula, Isabela Fernandes, and also Fabielle Russo. They share the first author because they work a lot on this job, on this project. So this is the experimental design, and we all in this work we used the Brazilian strain. So we were the first group using the Brazilian strain at this point. We also used the African strain, and also we used the yellow fever, which is another flavivirus. So we did the infections using both NPCs, neurons, and neurospheres, which are NPCs in a 3D model, and also we used brain organoids to see the behavior of the virus in these CNS cells. So we could notice that the virus could infect NPCs, and both NPCs and neurons, and could replicate inside vesicles. Here is a mature particle of the virus. The virus could diminish the size of the neurospheres. If we compare the first 24 hours, they have exactly the same size, and after 96 hours, we have the really strong impairment of the growing of the cells of the neurospheres infected by the Brazilian strain. They are much more aggressive than the African one. And also, we could notice that the migration of the cells is impaired compared with the control. Here is the one infected by Zika. So this could explain the impairment of the migration of the NPCs to make the cortical layer. This is our brain organoids. So when we try to see what the virus could do in the brain organoids, we could notice that this is 24 hours, the cortical layer, this is 96 hours after infection. So the size of the cortical layer is tremendously diminished, but not only the cortical layer is affected, we have also the ventricular zone infected by the Zika virus with loss of cells undergo apoptosis, apoptotic pathways. We also notice that we have autophagic pathways. And I want to call attention for this kind of cytopathic effect. So we notice that after infection, the nuclei of the size of the nuclei of the cells are big compared with the control ones. And also the nuclei seems with this empty aspect. So this was in vitro, but in 1971, Bell presented this paper with a similar results, but injecting the virus inside the brain of mice with a 2 days mice. So we have the same effect, but in vitro. So this is something to make us proud, because we have the same effect in vitro. We don't need to do this assay, in vivo assay, to see the same thing. And for last, we also have organoids, brain organoids made from cells from chimp, fibroblasts from chimp, from a biopsy in a chimp. And we'd like to test this brain organoid with the 2 strains of Zika virus. So we notice that in this case, when we have, it's not monkey, okay, chimps are non-human primate, but when we have these cells, the effect of the virus, the African strain, is the complete opposite compared with the Brazilian one, which is more aggressive in human cells. So we have better replication of the virus, of the African strain, of the African Zika strain, compared with the Brazilian strain, and also with the yellow fever. So this means that the strain that is circulating in Brazil since 1947, which is the first strain, Zika virus strain isolated, this strain is a kind of humanizated. So that's why we have this kind of phenotypic effect now, and we didn't have in the beginning. So more than one manuscript corroborated with our results, also the Stevens group. And we are still working on Zika virus, also the other disease that I showed you before. We are doing other approaches to understand a little bit more about the pathogenesis of Zika virus. And we are using this model to drug screening, as you all are using. So this is especially important. I want to call attention for the drug repurposing, which is the thing that we are doing. Most of us are doing this to using IPS platform to test drugs. So the drug repurposing we save at least 6 years. So when you discover a new drug, you have at least almost 7 years to discover something that really worth to start to go on. But here we have a kind of that valley with most of the drugs are just, we don't have we don't have academic support or money to proceed. So if you use, I don't know why this color is, okay. So if you use drugs that are already approved, we have 6 years less in this way. So we save time and then we save money. So it's about to do drug reproposing, we have just 6 years, and we have 3 million, more or less, dollars, the American dollars, we can save that. So this technology and also this platform are using for some of the most important pharmaceutical industries in our world. They have laboratories working with IPS model. So I just want to call attention of some labs here. They don't need any kind of presentation. So we are working with that, trying to see if we can find, we just work with drugs that are already approved to save time and money. But of course, we can use our platform to do to work with drugs, new drugs, as David also showed something about that. Okay, so I'd like to say thank you to the people from my lab. Most of the work that I presented here were done with some of these guys here, and also for some collaborators. I presented some data from Alisson Motri, Carol Marqueto from Rusty Gage's group, and also from people from USP and other collaborators that I didn't present any data with, and also for the funding agencies. Thank you.